Hi everyone. My name is Pranali and I work as a data analyst with one of the cancer hospitals in Bangalore. And I've done my master's in bioinformatics and I've had 5+ years of experience in the field of data analytics and biostatistics. Prior to this current job profile, I was working with a startup and I had my own side startup business, which was a service based company wherein I was providing data analysis to the research groups and labs across the world. So, and that's about it. Other than that, I would just like to thank LifeGenBio for giving me this opportunity to work with them and explore different arenas as well. So today we'll be talking about, it would be just a general introduction about bioinformatics and what is there or what is the scope of bioinformatics for the people who are trying to pursue their masters in the field. Also, I would like to cover up some basics about bioinformatics and what are the current situations that are happening or techniques that have been developed in the field of bioinformatics. So I think I should start now. So the contents of the workshop would be as such. So firstly, we'll talk about why bioinformatics and why is it needed. Then a brief introduction to the databases that are there or the tools and softwares that we are currently using in the field of bioinformatics. Then I'll talk a little bit about the three pillars, the three main pillars of bioinformatics, which are genomics, transcriptomics, and proteomics. As well as we are expanding our horizons in bioinformatics with machine learning and artificial intelligence as well. So I would also present a couple of examples as to in what aspect are we using machine learning and artificial intelligence and data analytics. And we will end the whole session with the situation of bioinformaticians in the current pandemic. Okay. So to start with it, bioinformatics is basically an application or a wedlock child of biology and IT. So you have the benefits of both the worlds. You can study biology and explore biology using the information technology techniques that are already established, or you can establish analysis, right? So again, you need to then biologist over the world, right? The one missing link between biology and technology is
Hello, ma'am. Sorry to
is
interrupt,
the
but your voice
interpretation
is
of that data because
hello,
of concept they can give us different. Yes.
ma'am. Your voice is breaking a lot. Can you please look
Yeah,
into?
is it still breaking?
Yes, ma'am. Yes.
Yes. Okay, just a minute. Okay, just a minute, let me try. Mm,
Okay, guys. Meanwhile, you can put in the chat box what are you doing currently. Do you have any questions prior to starting of the workshop? And do you? What is your current qualification, if any?
Stop sharing, recording. Okay, am I audible now? Is it better now?
Yes, ma'am. Loud and clear.
Okay, great. So I'll just share my screen again. So there is no issue. Okay, yes, yes. So should I start from the top again or do I continue?
Ma'am, please continue.
Okay, great. All right. So, like I was saying, there are three main aspects to bioinformatics. One is data generation and storage. Second one is data analytics, and the third one being data interpretation. So, why bioinformatics? Firstly, there's a very popular quote among us bioinformatics is that we have enough data generated for us to work on for the next 50 years, right? So there is a lot of data generation happened over the last 1 decade, and then we have enough problems to solve. Now the benefit that we have with bioinformatics is it is not it is very economical for everybody. It is very economically feasible because with wet lab techniques or wet lab validation, a lot of reagents, a lot of chemicals, a lot of manpower is needed to generate a whole experimental outlay, to follow up with the experimental outlay, and then to get the validation done. Whereas with bioinformatics, all you need is a good infrastructure. A good one computer should be fine to get your validation in place, right? So there is massive amount of data which all cannot be validated through wet lab techniques, and that's where bioinformatics comes into picture, wherein it allows you to generate a proof of concept or even validate your hypothesis to work on. So that is one of the major reasons why we need bioinformatics. Apart from that, again, we also need there's a need for efficient solutions. Every time it cannot you cannot always have to go through the whole rigorous process of validating something through a whole protocol of experimental designs to validate your hypothesis or to confirm your hypothesis, and that's where you need bioinformatics. Now, in bioinformatics, the general aim of bioinformatics is to develop, optimize, and parallelize algorithms or models which will allow us to help with our hypothesis or validate our hypothesis. Now, what we majorly do is take the raw biological data, which is generated by the biologist, interpret it, analyze it, and provide reports to the biologist in a way which they understand. Because a lot of biologist people are not very technologically friendly. They might generate amounts and heaps of data, but it's all French to them, right? So they are not really able to analyze the data that well or to interpret the data in a biologically sane format. That's where bioinformatics comes in, wherein it allows the biologist to interpret, visualize, and analyze the data in a way which makes biological sense. Now, there are a lot of parallelizations happening between the computer science and bioinformatics background people. So the basic differences between computer science and bioinformatics is proof of concept implementations are not just sufficient, right? In bioinformatics, you can't just have a proof of concept and say this is how it will work or this is how it might work. In bioinformatics, you have to walk your walk, right? So you have to perform all the simulations and analysis and do all the implementations instead of just providing a proof of concept. Apart from that, we also provide codes that are you need to develop an algorithm or a software which has a very simple GUI, which has a very simple functioning for anybody who is not technologically friendly to use. We also provide support for the code. So there are a lot of companies which have developed massive softwares like Strand Biosciences, right? Strand NGS. They have, or even Syngene for that matter. There are companies which have developed a lot of softwares and they have published those products into the markets, and they just provide support for those softwares, as in they help the biologist to run the software or to implement the software in the time or in the way that they need. Also, a bioinformatician cannot just be a one product hit. You cannot just have one algorithm or one code and be done with it, because again, it's a very booming field. There are a lot of problems which need to be addressed. There are a lot of problems which are arising every day because a lot of different types of data has been generated. So in order for you to be really successful in your field, you need to keep improvising. You need to keep trying different techniques to get the results that you want. Now, the aim of bioinformatics. So like I said earlier, the first aim of bioinformatics is to collect and organize the data that is already present. right? There's heaps and heaps of data that has been generated over the past few decades, with a lot of advances that are happening in the biology field or even the sequencing field, right? A lot of big data has been generated, and the current need is to organize that data. So there's a lot of data present everywhere, but accessing that data or extracting that data is a huge problem. So you need to have your database management skills in place, wherein it will help you to collect the data, organize the data, and put the data in a format where it is easily extractable for people. So that is currently being done through different databases like NCBI or Protein Data Bank or 3D molecular structures. You have Ensemble, which provides you with structures as well as sequences for the data, right? So that is the first aim of bioinformatics, wherein even if you don't generate new data, you can just work with the existing data that is there. The second one being data curation. Now, a lot of data that is generated does not necessarily be a very high quality data, or does not necessarily help you with a particular hypothesis or with a particular aim of yours, right? So you should be good at data curation. You should be able to curate your data, or at least extract data which is of good quality. Because even with something simple as sequencing, when it started, there were a lot of noise data that was generated with the whole sequencing techniques that are published right now, right? And over the time they have improvised on those techniques and found ways to develop and generate a lot of high quality data. So as a bioinformatician or as a analytical person, you should be able to curate that data and be able to extract good high quality data which will help you with your hypothesis or aim. So in that sense, you need to have your data mining techniques in place. And the third aim is to use these tools that are already present, or even generate or develop your own algorithms to help analyze the data, right? So these are the three main aims of bioinformatics. Now we'll just move on the three different pillars of bioinformatics, which is essentially your central dogma of life. The first one being genomics, then transcriptomics, and proteomics. So genomics is essentially the study of an organism's genome, wherein you study the entire genome on its whole and come to conclusions or help come to conclusions to the biologist. Now, genomics was first introduced by Thomas Roderick in 1987. There are two different types of genomics, one being structural genomics, another being functional one. So the structural genomics works more on the structure of your DNA rather than the functionality of your DNA, whereas functional genomics deals with the mutations and other other functional changes that happen to the DNA, which in turn affects the phenotype of a person, right? The second one being transcriptomics. Now, transcriptomics is essentially the study of RNA, which is basically the genes that are coded by the genome of a particular cell or an organism. Now, with the recent advances in sequencing, we have two types of transcriptomics that can be done. One being the bulk cell transcriptomics, and second one being single cell transcriptomics. I will get into the details of this later as we progress with the entire presentation. And lastly, it is proteomics. So proteomics basically allows you to study the protein profile of a cell. Now, protein is actually the end product of your entire central dogma. So any changes that happen in the DNA or RNA, or any translocations, deletions, or any kind of minute epigenetic changes as well that happen in your genome or transcriptome eventually go down and affect your proteins. So the most important pillar of your whole omic generation is the proteomic pillar, because the proteins that are at the end of the process that are expressed or not expressed actually have an impact on an individual or the phenotype of an individual. So that is how it works. So in order to even analyze or study these different aspects of omics, the first thing you need to know are the databases, as in the places where all the data is stored. Now, there are three different types of biological databases that we have. The first one being the structural one. The second one being the sequential one, and there are other databases which I'll talk about in detail. The structural databases are PDB, that is Protein Data Bank, SCOP, and CATH. Again, most of the structural databases majorly focus on the proteomes rather than the genome or the transcriptomes. So most of these structural databases have protein structures in them, which are generated using either crystallography or NMR techniques. Then you have your sequence databases. Now the sequence databases are in two types, one being the nucleic acid ones, which include your DNA and RNA sequences, and one being the protein ones, which include your protein sequences, right? So the nucleic acid ones are the general ones, which are NCBI, DDBJ, and EMPL. All these three databases are your primary databases, as in they have the sequences of all the proteins or the RNAs or all the transcripts and genes that are generated through multiple studies, and those are the ones that are stored in these databases. Similarly, for protein sequences, we have SwissProt, PREMBL, PIR, and ProCite. Again, these are the databases. You also need to remember, most of these databases have been generated but not updated, right? So NCBI keeps on updating. EMBL also keeps on updating their data on a monthly basis, wherein new submissions are given, new data is generated, all the data is first quality checked and then submitted to these databases. So similar is the case with Ensemble. With SwissProt, they have stopped updating their database for a very long time. So you need to be very particular about the kind of sources that you use for collection of your data or basically data mining, right? And the third type of database that we have is the literature or the curated databases. Now, when you talk about databases in general, like the sequential or the structural ones, they are not about any particular topic, or they are not focused on any particular aim, right? Whereas the curated databases essentially are focused and are working towards a particular hypothesis or a particular aim. So when you there's a database called GPCR database. So GPCR again is a receptor protein, cell receptor protein. So basically it helps in cell signaling of the entire cellular state. And there is a database only and only denoted towards GPCRs, their sequences, their structures, their functionalities, their cell-cell interactions. So those are the databases which are called the curated databases. There are multiple examples of such, wherein there are databases which only focus on proteins which help with proliferation, or only focus on proteins which help with adhesion. So there are multiple curated databases which help and support a particular cause or an objective of a project. So that way, it is a great initiative. So say you want to work only, so for me, like I work with glioma patients, that is brain tumor patients. So I have generated a database which only focuses on glioma data sets, which only has data sets where the patient data is a glioma patient data, right? So those are your curated databases. Apart from that, you also have literature databases, wherein PubMed or PubMed Central, these are your literature databases, wherein all the literature reviews or research articles that have been published are stored in a particular database, that is your literature database. So again, when you are looking through a literature database, there are multiple factors you need to take into consideration, as in when was a research article published, right? So you do not want to look into, given the current boom that has been happening in the whole field of biology as a whole, you do not want to look into articles which are prior to 5 years, I would say, right? Because all the technologies are really adapting new techniques, adapting new approaches to solve the existing problems. So anything that is outdated is not something that is going to help you with your hypothesis or the aim of your experiment. So again, with literature databases, you have to be very careful as to what kind of literature are you looking at, and how is it going to help you with your hypothesis. So these are the basic databases that are there, which will allow you for data mining. Now, there are major, so data mining in itself is a entire uh, arena of different topics, wherein right? So data mining in itself can be a different entity or a different project altogether, or a subset of bioinformatics, right? So data mining includes various steps, which I'll be covering now. The first step being data cleaning. So data cleaning is essentially, like I said, a lot of data is generated, and even today when we generate data, the specificity or the accuracy of the data is not 100%, right? The technology that has been currently being used for any kind of data generation or sequencing generation is not 100% accurate. So it generates a lot of noise, a lot of garbage along with your actual data. So the first step, the very first step of data mining should be data cleaning, as in only extracting the amount of data that is actually good quality and which will help with your hypothesis. Now, the data cleaning with respect to literature, like I said earlier, you want to focus only on data which is recent. You want to focus on data which is new, or which is actually adapting to the newer technologies. If you're looking into doing a review, say for sequencing, you wouldn't want to go back to Sanger sequencing and try to do what they have done, right? You would want to adapt to the newer sequencing techniques that are there. Similarly, with data analytics, when a lot of sequencing or RNA-seq data is generated, a lot of it is background noise. A lot of it includes data which might be mismatched or incorrect. So the first step we always do is data cleaning. The second step is data integration. So initially when the data was generated, it was generated using only 5 samples, 6 samples, 7 samples. Compared to now we have an individual data which will have up to 10,000 samples or more than that, right? So the first step, the second step was always data integration, as in when you do a meta study or meta analysis, you would always want to include multiple sources to help your hypothesis. In statistics, it's always a rule to include multiple sources. You cannot prove your hypothesis based on a single source. So in order to do that, you do data integration, that is combining of multiple data sets. Now, while you're combining a lot of data sets, you need to take into consideration the technologies that were used for different data generation, the kind of batch effects or the kind of noise that was introduced into the data while data generation. So integration is again an important step. The third step being data selection. Now, as we know, let's take a hypothetical case of a particular disease. Let's talk about diabetes. Okay. So when you want to see what are the particular genes that are expressed in a diabetic patient versus a normal patient, right? The first thing you want to do is look at the entire gene structure or the gene profile of a diabetic person as well as a normal or a healthy person, right? Now, when you try to do that, there are 20,000 protein coding genes that have been annotated in the human genome, right? So not all 20,000 will be differentially expressed or will have different profiles in a diabetic or a normal person, right? So you need to select out a particular gene which is of your interest, which you know is dysregulated or is deranged in a particularly diabetic patient. So that is what is data selection. You have thousands and thousands of samples that are there. Not all thousands of samples are going to help you with your hypothesis. So you need to be careful about selecting your data. Okay. After that, you have data transformation. So essentially, data transformation is basically you kind of arrange or transform the data which is interpretable, right? So you perform logarithmic transformations or you perform some other kind of normalization techniques to your data in order for you to analyze the data. The fifth one being data mining for knowledge discovery. Now this is where machine learning comes into picture. So you apply particular techniques to mine data or mine patterns in your data. So you know that particular gene is dysregulated in cancer patients, right? So you look for that particular gene across all people. You mine that particular information from multiple data sets, right? That is basically data mining for knowledge discovery. And then pattern evaluation. Now you have established a pattern. Say you have established that BRCA gene is upregulated or downregulated in breast cancer patient. This is the pattern you've observed across, say, 10 patients, right? Now you want to evaluate this pattern to a larger population, in larger populations, right? So that is pattern evaluation, wherein you have established a particular design or a particular pattern that you've seen or observed in a smaller population. Now you evaluate the same pattern across a larger population. So these are the basic steps of data mining. Now in genomics, again, like I said earlier, the genomics, transcriptomics, proteomics, and metabolomics. Metabolomics is essentially the study of metabolites that are produced in a genome, right? So all of these different pillars, when they come together, they form the phenotype of a person, right? A height of a person, weight, eye color, hair color, skin color. These are all affected by different omics that are there in your entire cellular structure, right? So when you talk about genomics or transcriptomics or proteomics, any of these omics, the first thing you want to see is how is it affecting the phenotype of a particular individual. Okay, so the steps for analysis are pretty simple. Just to explain them broadly, the first step is always generation of data, right? So if you are a bioinformatician, mostly you skip this step because the data generation is not a job profile or a job for you. Either you already take the publicly available data sets to work on, or a biologist will generate the data for you to work on, right? So most of the established public data sets that are there. So for cancer, you have TCGA. And for any other disease, you also can download data from GEO database, which is a subset of